Thank you, Dr. Chance, for the introduction remarks. Good morning, everybody. My name is Giri Gokulrangan. I'm a research staff member here at the Case Center of Proteomics and Bioinformatics. Once again, a warm welcome to one and all for our symposium here titled Understanding Protein Complexes, Interactions, and the Interactome. We'll kick off the talks today with an internal speaker, our own Dr. Rob Ewing. He's an assistant professor here at the Case Center for Proteomics, and also he holds a secondary appointment in the Department of Genetics. He's the director of Bioinformatics at our CPB Center, and his previous appointments include senior research scientist at Insight Genomics, and also he was the group leader at MDS Proteomics in the Bioinformatics division before coming here. Rob also is a co-founder of Infochromics. It's a bioinformatics startup. The research focus areas for Rob include wet lab approaches for mapping protein-protein interaction, and also computational approaches for predicting protein-protein interactions. It's a pleasure to start the day with his talk. Dr. Rob Ewing.
So, the next 20 minutes or so, I'd like to first sort of introduce the sort of area that we're going to be talking about and follow on really from what Mark said, and also tell you about some of the projects that are ongoing in my lab within the center. So Mark sort of alluded to this, the fact that we can really think about, protein interactions as occurring at different scales, all the way from I don't think the pointer's working. Has anyone got another pointer? Fantastic. Thank you, Jeff. So now I have to coordinate two things. That could be tricky. So we can think about interactions occurring all the way from individual interactions that we'd study at the structural level through high levels of organization of interactions into complexes and networks, and then ultimately looking at the full complement of interactions in a complete biological system. And of course, we're going to be using different technologies to study interactions at those different levels. And so when we were thinking about what we would schedule for today, we really wanted to provide a fairly in-depth focus, mainly in this area. But as Mark said, ultimately what we'd like to have a full sort of structural information about individual interactions at a global scale as the ultimate goal. So in thinking about why we'd want to study protein interaction networks and the kind of information we can get from interaction proteomics, to my mind, it really sort of boils down to three important areas. Firstly is being able to assign function through association. So if proteins or other biomolecules associate with one another, then that may tell you something about their respective functions. The second is the topology of the networks itself, so who's interacting with who. And then thirdly, I think an area that will become more and more important is reconfiguration of complexes and networks in response to perturbations within cells in different states. This is our current favorite example of this, and I think it's a really nice example. This is a pictorial of the canonical Wnt signaling pathway. And so, to cut a long story short, essentially in the presence of Wnt, you get this complex, which is key to the transcriptional response, the Wnt transcriptional response. And in the absence of Wnt, you get the so-called destruction complex, which is, if you like, reconfigured in terms of both the modifications and the interactions. And so the topology of this complex is absolutely critical to this, to Wnt biology. And I'll tell you a bit more about Wnt biology later on, because that's an area that we're interested in. So I'm going to tell you about several things today. I'd first like to start off by telling you about something that I've been involved with over the last few years, which is generating a a human interactome set where we took a fairly large number of human proteins and mapped interactions for those proteins. I'll then tell you about how we're using that data set to do more focused work in the Wnt regulatory network. And then I'll come back to talking more about methodology and tell you about two other projects in the lab. First is a computational project where we're looking to computationally classify baits according to technologies that they would be appropriate for. And secondly, how we're using knock-in cell lines for interaction proteomics experiments. So first, just a note on technology. What I've done here is really just provided schema, really, of perhaps the two key technologies in terms of high-throughput interaction proteomics. So there are lots of ways of measuring protein-protein interactions, but the two methods that are provided most of the high-throughput data are outlined here. So on the left, the yeast-2 hybrid. Probably most people are sort of familiar with this, but essentially it's a very elegant system that was originally conceived when it was noted that you could take a transcription factor and actually split that transcription factor's functionality you could actually split the activation domain and the binding domain of a transcription factor and then reconstitute the overall activity of the transcription factor by bringing those two things back into physical proximity. And so you can use this by fusing the activation domain to one protein and your binding domain to another protein. You can actually assay whether those proteins interact. And as we've seen over the last few years, this technology has been applied at large scale. What the technology really tells you, and this is a key point, is about binary interactions. It's really testing whether one protein interacts with another. And that contrasts really with our other technology. This is our sort of favored method for looking at protein-protein interactions, which really tells you about protein complexes. So affinity purification mass spec, very briefly, you take a target protein of interest, and you express that protein with an epitope tag, typically in cells, and then you use that epitope tag to purify the bait protein. And if your experiment works, then hopefully you'll also be able to identify or purify some of the binding proteins to the bait protein. So these are represented here. And then, of course, we're using mass spectrometry to actually identify and potentially quantitate these potentially interacting proteins, and then hopefully assign them into protein networks. So I'll talk more about APMS as we go on and provide you more detail. So I want to tell you about some work we did a few years ago whilst I was in Toronto in a proteomics company. This actually was a data set that was generated for it's a human interaction proteomics data set. The data was generated in a fairly uniform manner in a sort of industrial process, so I think it represents quite a nice data set for further analysis. The bottom line is that we eventually published data for around about 350 human proteins, human baits, and that corresponded to around about 1,000 individual affinity purification experiments. So we came up with a sort of a higher confidence network of around about 6,500 interactions corresponding to over 2,000 distinct proteins. The workflow that we used is broadly outlined here, and I've told you a little bit about this, so I won't tell you too much. I just want to point out a few things that I'll come back to later. The first is that, of course, you have multiple choices of how you actually express your protein, and all this data, the Pathmap data set, was all generated using transient expression. What I'll tell you a little bit about later is a technique that we're using now in collaboration with John Wang's lab in genetics, where we have knock-in cell lines rather than transient. I'll tell you more about why we're doing that later. So I just want to tell you, I just want to point out a few features of the Pathmap dataset that are relevant to today. The first is that in affinity purification mass spec experiments in general, one of the big challenges is actually the fact that your affinity purifications largely look like this, where your bait and your presumably putative interacting proteins are there, but you also have sets of proteins which are non-specific for your bait, maybe binding to the resin or whatever kind of purification you used. So in an ideal world, we would have this scenario where we don't have any of these gray proteins here, but we don't. We have this. So what we did with the Pathmap data set to try and identify these proteins and control for them was to do a fairly large number of control affinity purification mass spec experiments. And these essentially correspond to, so the way we did these was to express the vector alone. So we're not expressing a bait, we're just expressing the tag and the vector. And then we go through the whole procedure of affinity purification mass spec. And the reason really that I want to mention this is that I think these kind of data are actually critical to interpretation of affinity purification mass spec experiments. We note that, in fact, these experiments are very reproducible. So you see the same proteins over and over again. They're not all just mass spec contaminants like the keratins. Some of them, in fact, quite a lot of them correspond to this, this complex here, the methylosome, in our system. The other thing I would say is that I think the importance of these data and the utility of them is reflected in an effort that actually Alexei and Anne-Claude, who will talk later, are spearheading, where we're going to collect some of these data and share them with the community. So the second thing I'd like to point out about APMS and our Pathmap dataset is that you can actually, this really, if you like, is, I think, the one strength of affinity purification mass spec, in that you can generally purify, at least for certain complexes, you can purify a large number of interacting proteins with relatively few baits. So this is an experiment that we did, or a series of experiments that we did with human baits corresponding to the proteasome. And we used 5 baits and actually managed to identify a significant fraction of the overall set of subunits. So I think this really, so actually I should say over here, these larger nodes in this graph represent the 5 baits, and then these smaller ones represent the preys, and all the red and yellow ones are proteasomal components. So this, I think, is the real strength of APMS in looking at protein complexes. So in terms of utility of the Pathmap dataset going forward, we really think in two very broad sort of areas. The first is obviously discovery. So a lot of the interactions are novel, and so the question would be, can we analyze the data and identify interactions which are perhaps important for disease? In the original data set, many of the baits are in fact associated with human diseases. We're particularly interested in colon cancer and other cancers with respect to Wnt signaling, so I'll tell you a bit more about that in a moment. The other thing to say is that some of the data, at least half of the, or about half of the data, is actually replicated, and so that means that we're doing multiple experiments for each bait. Some of it's quite highly replicated, so for 17 different baits, we have 4 or more replicates. So we like to think that we can also use this data, this data set for further data analysis, method development, and benchmarking. But I won't tell you too much about that today. So one area I'd just like to talk about briefly, this is really a project in sort of conception rather than fruition, if you like. So we've been looking at this, and so we're really interested in the Wnt signaling regulatory network, and we think that we can make a contribution to understanding how the cellular network regulates Wnt signaling. And one reason, or 3 reasons why we're really interested in Wnt signaling, as many of you will know, the Wnt signaling is a highly conserved signal transduction pathway. And so there are some very nice tools and reagents, really, and cellular and biological systems for studying Wnt signaling. The second is embryonic development. Wnt signaling is critical to embryonic development, and any perturbation of Wnt signaling results in these sort of extraordinary phenotypes. This is in Xenopus. And thirdly, importance for disease. So I guess the best example is probably colorectal cancer, but more recently, several papers have pointed out interesting associations between canonical Wnt signaling and other conditions such as Wilms tumor and schizophrenia. So we think there's going to be more discoveries, and that the sort of central importance of this pathway to disease will become increasingly evident. So this is, if you like, is a sort of grand conception of the way we're thinking about how we might tackle this. So on the left here, what I've done is I've put a sort of a subset of the both the canonical Wnt signaling components or Wnt signaling components with some of their interactions, and just demarcated some of the proteins that we've actually, the ones in red are the ones that we have some data for in the Pathmap data set. So we see the Pathmap data set really as a foundation that we can use for data mining to actually pull out novel regulators. Now, what we're specifically interested in is really using different kinds of data and building out this model. And the reason we're really interested in that is that in Wnt signaling, essentially like other signal transduction pathways, you get a transcriptional response. And it's been noted that the targets, if you like, of Wnt signaling, or at least some of the targets of Wnt signaling, are also actually components of the pathway and regulate the pathway itself. So we see this as an opportunity by using different kinds of data of actually picking up on Wnt targets that are both components of the pathway. And to do that, we see really four critical kinds of data or types of data. The first, obviously, is protein complexes and networks. We have some of that data already. But then we're thinking of, if you like, a fairly careful and select integration of other kinds of data. So functional interactions. There's been a number of functional screens for Wnt regulatory components over the last few years, which have revealed some interesting data. And of course, more transcriptional data, gene expression and motif information can tell us about this end of the pathway. So here's a little bit more detail. And this, if you like, is our platform, or we hope it will be our platform for discovery of Wnt regulatory components. As I say, what we're really starting with is our interaction proteomics data. We want to supplement those interaction proteomics data with functional assays. So the idea here is that you could have a physical network of interactions, but that might not tell you anything about the functionality of those proteins. So what we want to do is to establish a reasonable throughput system where we can actually inject target proteins into Xenopus embryos. And the big advantage about this is that when you inject either antagonists or agonists of Wnt signaling into Xenopus embryos, you get these very clear phenotypes. So we're hoping that we can both identify physical interactions and functional ones using this system. The other parts of our platform really are computational. So as I mentioned before, we're seeking to integrate different kinds of data. And then sort of data mining. And I just want to very briefly tell you about some of the sort of earlier, our sort of initial efforts at doing some of this data mining. So this particular protein, ETAA1, is Ewing's tumor-associated antigen. There's no connection. It just kind of popped out like that. So, but anyway, we identified ETAA1 as a potential interactor with some of our Wnt-related baits, which we thought was interesting. But then we went to look, then we were thinking, well, can we see whether ETAA1 is also a Wnt target? And one way to look for Wnt targets is to look for these so-called TCF sites, which are the regulatory sites in the promoter. And we did an analysis across all of the genes in the genome and found that ETA1 has a very skewed distribution for the number of TCF sites. And in fact, when you look at those TCF sites, this is just a sort of cartoon of the promoter region of ETA1. You find that these are the TCF sites here, at least 3 of them. They're actually conserved. So we're quite excited by this, and we think that these kind of targets will be good to go back and test using our interaction proteomics and also our functional assays. So what I'd like to do for the last little part of the talk is tell you a little bit more about the APMS workflow and what the sort of refinements that we're making in the lab that we think will improve our ability to really identify good protein-protein interactions. And I'm going to focus in 2 areas. The first is a computational area where we've been trying to come up with more of a sort of a rational way of bait selection, if you like, and I'll tell you about that in a moment. And then the second, as I mentioned earlier, are these knock-in cell lines that we've been using for APMS. So, okay, I'm going to skip over that. So, in thinking about sort of bait selection and classification, our initial, our sort of conception is sort of cartooned over here. So if you imagine the full complement of protein interactions in a sort of multi-dimensional space, then you could potentially in the future map your different technologies like affinity purification mass spec or yeast-2 hybrid or any other technology to that space in terms of the interactions that you're able to cover in that space. So no technique is going to be good for everything. Different techniques may have some overlap. They may have unique areas as well in this space. And so one thing we were wondering is whether, you know, for any given protein that you might want to use as a bait, can we sort of predict whether that protein will make a good APMS bait or perform in yeast-2 hybrid studies, or indeed whether it's just completely intractable to current technologies. Sudipto Saha, who's a postdoc in my lab, has been looking into this problem, and this just sort of outlines the formulation, the initial formulation that we've come up with. The idea is really quite straightforward. It's simply to ask the question of whether for bait proteins, can we use bait proteins or bait features of bait proteins to predict whether those baits will be successful? And what I mean by successful is whether we'll find good preys using different technologies. And so the idea is to build a statistical model where for each bait we take the set of features, and these are some of the features that we've been looking at, to build a statistical model to actually predict success or failure according to different technologies. And so Sudipto's model actually performs reasonably well. If it was 100% accurate, that would be fantastic, but it's certainly better than random. And I'll show you exactly how we can use this kind of data, both to study the features which seem to determine whether an experiment will be successful, but also in a way to sort of predict which baits to use. So what you're looking at here is for each row in this heat map represents a term or a feature, and some of these are redundant because different sources of annotation tell you about the same thing. But essentially, what we've done here is pulled out all of the features of proteins which are over or underrepresented according to the different data sets, yeast-2 hybrid or affinity purification mass spec. And you can, once you've got this, assembled this data, you can then pull out areas of interest. And I think these are probably 2 of the more interesting areas where you may have one technology which works well and the other technology that doesn't work well, or vice versa, or neither technology that works well at all. And in fact, there's no surprises. They're not all listed here because it's a much longer list, but there's no surprises that membrane proteins actually are quite intractable to both yeast-2 hybrid and affinity purification mass spec. I want to tell you very briefly about, this data is sort of hot off the press, so, but I want to tell you very briefly about more in-depth analysis that Sedipto's been doing looking at peroxisomal interactions. And peroxisomal interactions are in this set here where they don't really work well with APMS, but they do with yeast-2 hybrid. So what we've laid out here are really, if you like, subsets of the complete interactome. And there's really sort of 2 points to be made here. First is that, as I said, yeast-2 hybrid performs much better with this subclass, if you like, of the interactome in terms of the baits that are annotated as peroxisomal baits than does APMS. I'll just say, actually, that the baits are represented by these large nodes and the praise by the small nodes, and the red ones correspond to peroxisomal proteins. The other point to note is that we can color the baits according to their predicted score of how well they're going to perform. And we find that does quite a nice job. So all of these baits here are not predicted to actually find praise or to work with the yeast-2 hybrid platform. And that turns out to be quite a good prediction, and similarly with APMS. So the next step is really to ask the question, we're at the point of wanting to ask the question of whether we can use this sort of bait classification score to better enable us to select baits in our Wnt signaling network that will make good affinity purification mass spec baits. So just the last couple of slides, I want to tell you very briefly about another area of work in the lab. So as I said earlier, there are different ways of expressing your tagged baits in cells, and we are exploring an area. We're exploring the possibility that we can use a knock-in strategy to generate cell lines, and this is a collaboration with John Wang, who's in genetics. John came up with a very nice and convenient way of generating tagged cell lines where he can knock in flag tags into different human cell lines. And he showed that you can use these knock-in cell lines for different purposes. You can do sort of visualization. If you look at the sort of Western level, you get the right interactions and so forth. He's generated a number of these lines. What we wanted to do was take one of those lines, and it's a line with a tagged MRE11 gene. Now, MRE11 is a fairly well-characterized protein that forms a complex, the so-called MRN complexes, which is really key for DNA damage and other sort of transcriptional and nuclear functions. And there are really, this is all sort of very well known, very well studied, which is kind of why we chose it. But there's a couple of very well-known interactors of MR11, Nibrin and Rad50. And so we've done actually a reasonable number of pull-down experiments using this cell line, and we found very nice consistent results as compared to our control, where we can find MR11 and Nibrin and Rad50. So that's sort of highlighted on this interaction map here. The complex is very, very nicely pulled out. And we can also hopefully use this for identification of some other potentially more novel interactors. So I think we're quite excited by the utility of these knock-in cell lines, and we think they're going to be a good supplement to our usual strategy of doing transient expression, because, of course, you conserve the endogenous promoter and transcriptional regulation when doing this. So I'm just going to summarize very briefly. I've told you about some of the technology and some of the ways that we want to use that technology, in particular to study Wnt signaling in the lab. I'd like to thank a number of people for their contributions to this work. In my lab, Jing and Yelena are really doing all of the experimental work. Sedipto is leading the bait prediction project and is assisted by other computational people in our center, particularly Parminder and Gaurav. I'd like to thank our moderator, Giri, for his excellent mass spectrometry. And also, I'd like to mention this collaboration with Mehmet Koyatürk, who will speak later, and 2 undergraduate students who are doing the network reconstruction. And then finally, in genetics, we've been well assisted by John and Zanwen Du, who generate the knock-in cell lines. And then finally, Keiko is our partner for doing the functional assays in Wnt signaling.
You have room for one quick question from the audience.
So Rob, have you ever compared your endogenous tagging with stable and with transient transfection, and do you find anything significantly different in the systems that you've looked at so far?
Yeah, we have compared it, and we haven't, I wouldn't say we have statistically solid enough data to say that overexpression or the knock-in approach, to really say one way or the other whether one is giving us one thing or another. I guess the other thing we've looked at is actually the level of expression, and we've only really done this for one example, but Jing took MR11 and did sort of just a regular over a transient expression and looked at the levels of MR11 expression by Western. Actually, they weren't so different from the knock-in, right? So, which I was a little bit surprised by. I thought initially that overexpression using a constitutive CMV promoter would just, there'd just be loads of it in the cell. I think these different approaches are going to be quite complementary. I think the nice thing about the knock-in is that you are preserving that endogenous regulation in terms of transcription, so that opens up new avenues. On the other hand, to get to some of the weaker interactions, you actually might need a higher level of expression with the risk that you could have false positives. So I think they're going to be quite complementary.
Thanks, Rob. Thanks for your